Welcome everyone, and thank you for joining us today for our webinar titled Implantable Infusion Pumps, Insights for Your Next Animal Dosing Study. This is Martin Hess from Inside Scientific, and I will be your host for today's event. Our session today is sponsored by Durect Corporation and will focus on features, benefits, and applications of implantable pumps for drug dosing studies in preclinical research. Our speaker today is Mr. Jose Gadea, who has over 15 years of technical and marketing experience with ALZET Osmotic Pumps and currently holds the position of Senior Product Marketing Manager for ALZET Pumps and Ancillary Products at Durect Corporation. Prior to Durect, Jose worked as a therapeutic sales specialist for ALZET Pharmaceuticals. He also has over 5 years of combined academic and biotechnology research experience in the fields of cancer immunology, cell and gene therapy, infectious diseases, and drug delivery. During the registration process, we asked you to submit one question for our presenter pertaining to the use of implantable pumps. We have collated all your responses and used this information to generate the content for today's webinar. With well over 500 scientists registered for today's session, we had lots of information to put together, and we do plan to use the full 60 minutes allotted for today's session. So with that in mind, let's move right along into our presentation.
Okay, thank you again, Martin, for the nice introduction. And hello, everyone. Thank you for joining our webinar today. I would like to start by showing you what we're going to cover today. And I will start by an overview of laboratory animal dosing options, followed by a brief introduction and history about implantable infusion pumps. Then I'll cover some features, benefits, and applications of the most common implantable pumps used in research. And along the way, I will be presenting helpful tips and suggestions for planning your animal dosing studies. I'm also going to be identifying and clarifying the common misconceptions surrounding the use of ALZET pumps in research, actually all ALZET pumps and iPRECIO. And as Martin mentioned during the introduction, the content of this webinar was specifically developed based on the questions that you submitted during the registration process. And if any questions were not answered during the presentation, we'll have some time for a question and answer session, and hopefully we'll get those questions answered at that point. So let's get started with the webinar. And as you know, the administration of substances to laboratory animals required careful consideration and planning to optimize delivery of test agents, maximize animal welfare, and also ensure the scientific validity of experimental results. Different animal dosing options are available for researchers, and appropriate options ultimately depend on the many experimental factors actually, including animal model, the length of the study, the nature of the material being administered, and just only to name a few. And the administration of a single dose is not really a problem. However, when treatments must be given repeatedly over time, this really presents some challenges. So it's important to have a general understanding of the advantages and limitations of the various dosing options. Injections are the most common method for dosing laboratory animals since they're readily available in the lab. And they're relatively simple, quick, and inexpensive. However, this method is not optimum for some drugs, especially those that have short half-lives and require multiple dosing. In these cases, injections can be very time-demanding and lead to significant animal stress due to frequent handling and repeated needle pain. They also result in inconsistent drug levels. Dosing by food or water is also simple, quick, and inexpensive. However, this method cannot be used for many drugs, particularly those that are insoluble in water or those that change the palatability of food or water. And this method also leads to inconsistent drug levels, since dosing is depending on the animal's drinking and feeding patterns. Oral gavage is also perceived as simple and inexpensive. However, this can be a very challenging procedure and can induce high level of animal stress or even mortality due to the potential tissue trauma during needle insertion. Biodegradable pellets are also available as a dosing method for some drugs, and this can be easily implanted with a trocar. However, many compounds cannot be formulated in a pellet, and others require special customization, which significantly increases the cost of the product. Catheter infusion is commonly associated with bench-mounted pumps, which provide increased control over the dose and enable administration of high volumes. They also allow easy access to the tech solution, which can easily be changed when drug stability or solubility are of concern. However, this method requires costly equipment that also requires continuous maintenance. Since the external catheter is required, there is an increased risk of clotting or disconnection. And this procedure also restricts animal movement, which required single animal housing. So ultimately, all of this leading to increased animal stress. Ambulatory infusion pumps provide similar benefits in terms of dose control, administration of high volumes, and easier access to test solutions for replacement. However, this method also involves costly equipment and the use of a catheter to hold the pump in place, and similar animal welfare disadvantages as other external pumps. So on the other hand, implantable infusion pumps are a very convenient method to deliver drugs in biomedical research. They provide dose control, which also improves drug efficacy and reduces side effects. They also provide significant animal welfare benefits over repeated dosing and external pumps, all of which results in better research data. One disadvantage of implantable pumps is the requirement for surgery to implant the pump, which is obvious. Also, drugs must be formulated as a homogeneous solution for efficient delivery. There's also a higher cost depending on the alternative method being considered, for example, injections or other cost-effective methods. The ALZET Osmotic Mini Pump and the iPRECIO programmable pumps are the most common implantable pumps used in research today, and I will focus my attention to these technologies during the rest of my presentation. But before, I'd like to provide a brief history about these implantable pumps. ALZET pumps have been around for a long time. They were first developed and commercialized in 1977 by ALZET Corporation, which is a pioneer in drug delivery. Direct Corporation, a specialty pharmaceutical company with proprietary drug delivery technologies, acquired the ALZET product line in the year 2000. And now we manufacture and distribute ALZET pumps worldwide. The iPRECIO pump was most recently introduced during the last decade by PrimeTech Corporation, which is a manufacturer and distributor of medical and analytical science products based in Japan. The first iPRECIO prototype was developed in 2007, and new and innovative product improvements have been made since then. Direct established a distributor agreement with PrimeTech to distribute iPRECIO pumps in North America in the year 2000. So ALZET pumps are essentially miniature infusion devices used for continuous dosing of unrestrained laboratory animals. They allow chronic administration of experimental agents at controlled rates. The ALZET pump has become a very useful tool in preclinical research for continuous delivery of many experimental agents, including chemotherapeutics, enzyme inhibitors, small molecules, proteins and peptides, nucleic acids, and many more. It is particularly useful for continuous delivery of compounds with short half-lives, which are very difficult to deliver by conventional dosing regimens. And their use has been cited in nearly 17,000 publications. Here are some features of the ALZET pump that are important for their use in preclinical research studies. The smaller pump models are optimal for implantation in animals as small as mice, which is the preferred species for in vivo research. Although it's primarily used in rodents, the ALZET pump can also be used in dogs, monkey, birds, fish, reptiles, and many other different species. They are a very reliable method for drug delivery. This can be validated by nearly 40 years of research use and the impressive number of publications in the literature. Complete implantation of the infusion device eliminates the need for repeated handling and also allows the animal to move freely around their cage during the infusion period. This reduces unnecessary stress and experimental variables which can interfere with the study results. So in general, ALZET pumps offer a convenient alternative to injections and other repetitive dosing methods, especially when used in studies requiring chronic administration. The pumps are also cost-effective and easy to use, as we will learn later in the presentation. They have a simple design that does not require software or programming to operate. But one of the greatest features of ALZET pumps is their ability to reliably deliver compounds at a constant rate. So let's review some of the benefits of continuous administration. So in addition to reducing animal handling and enabling automatic dosing of laboratory animals, continuous delivery can improve the ratio of therapeutic effects to toxic effects. This is critical in preclinical research since many drugs are frequently restricted by their low therapeutic index, where side effects occur at relatively low doses while maximum therapeutic activity demands higher doses. For some agents, continuous infusion is more efficacious and with fewer side effects compared to administration by injections. Continuous administration can also produce savings in the amount of compound utilized in research studies. This benefit has greatest potential for compounds that show fast clearance rate in vivo and for which frequent dosing is required to maintain effective levels in plasma or tissues. This savings is also important in early stages of preclinical research investigation, when compounds are very expensive to produce or sometimes even available at very minimal quantities. The benefits of continuous administration are evident on this graph, showing plasma levels after drug administration by daily injections compared to continuous administration by ALZET pumps. A therapeutic dose can be maintained throughout the entire experiment using ALZET pumps, as you can see in the blue line here in the middle of the graph. On the other hand, injections will result in peaks and troughs. At the peak, you will be above the therapeutic level, resulting in drug overdose, unwanted side effects, and drug waste. And at the trough, you will be below the therapeutic level, which results in loss of drug effect due to underdosing. So let's now review the pump's mechanism of operation and see how it works to enable continuous delivery of experimental solutions. The ALZET pump has a cylindrical shape and is composed of 3 concentric layers: the inner drug reservoir, the osmotic layer, the outer rate-controlling semi-permeable membrane. The flexible reservoir is composed of a synthetic elastomer, which is chemically inert to most drug formulations and is impermeable to the material in the surrounding osmotic layer. The osmotic layer in the middle contains a high concentration of sodium chloride that provides the osmotic driving force. And the outer rate-controlling membrane contains a proprietary formulation that allows selective water permeability. An additional component of the pump is the flow moderator, which is a 21 gauge tube with a plastic end cap, which prevents random drug diffusion and ensures that the delivery profile is osmotically controlled. So here's how these parts work together to provide constant delivery. When placed in an aqueous environment or implanted in an animal, water enters the pump across the semi-permeable membrane into the osmotic layer. This hydration process causes expansion of the osmotic layer and puts pressure on the flexible reservoir, forcing the drug solution through the flow moderator and out via the delivery portal at the tip of the pump. And since the outer membrane is rigid, the volume of water entering the pump is equal to the volume of drug solution that is released. The delivery rate is dependent on the membrane permeability of the system, and various formulations have been developed to provide researchers with a wide range of delivery rates to choose from. So in fact, there's a total of 12 pump models that are available in 3 different pump sizes, that, as you can see here in the graph, the 100 microliter sizes, the 200 microliter sizes, and the larger 2 milliliter sizes. Infusion durations range from 1 day to 6 weeks, which is the longest duration pump, the 2006. Note that nominal specifications are listed on this graph as it is on all other marketing materials. Individual lots of pumps will vary within this, from this target within acceptable limits, and I will discuss the importance of this concept later in my presentation. In general, ALZET pumps are relatively easy to use, and I have summarized the simple steps here on this slide. The first step involves the preparation of the drug solution in the corresponding vehicle at the required concentration to deliver the desired daily dose. Then the pumps are filled with this drug solution, then primed so that they are ready to deliver at the specified release rate at the time of implantation. Then the pump is implanted in the animal, and once implanted, the pumps will deliver a constant automatic dose throughout the study period or the specified duration of the pump. Choosing the right pump for your study is based on many different factors, such as animal size, route of administration, duration of infusion, or drug solubility. In general, we recommend using the smallest pump possible for a given duration. Then you will need to determine the drug concentration required to achieve this desired daily dose. If the calculated concentration is within the solubility parameters of your drug, then the smallest pump for a given duration is your best option. However, if you find that the required concentration is too high and beyond the solubility parameters of your drug, then you'll either have to find an alternative vehicle to improve solubility of your drug, or consider a pump with a larger reservoir volume or a faster infusion rate, or you may even need to resort to a combination of both options. With respect to the agent that can be used with ALZET pumps have actually been successfully used to deliver a wide range of compounds, and that includes peptides, hormones, nanoparticles, steroids, growth factors, and just to name a few. One common misconception about ALZET pumps is that researchers assume that they are not optimum for delivery of large-size compounds. This misconception originates from the belief that drugs are released through the outer membrane of the pump. But as we learned earlier, the delivery rate of ALZET pumps is controlled by the water permeability of the outer membrane, and the drug solution is released through the exit port at the tip of the pump, which is a 21-gauge tube, as I mentioned earlier. Therefore, the delivery profile is independent of the compound's molecular weight or chemical properties, and compounds of various molecular configurations, including macromolecules, can be successfully delivered at predictable rates. There are 2 very important criteria that the compound must meet to guarantee success when using ALZET pumps. First, the compound must be stable at 37 degrees Celsius for the duration of infusion. And we say 37 degrees because we assume that you're working with mammalian species. If the compound stability is not known, perhaps we can help by searching our technical resources and publications for that information. Otherwise, you may need to perform a stability test to determine if your compound will retain biologic activity for the duration of infusion. This information will be critical for choosing the right pump for your study. For example, if you plan to do a 4-week pump study using a 4-week pump, but your compound is only stable for 2 weeks, you could use a 2-week pump and replace it with a new pump to achieve that entire 4-week duration with a biologically active compound. The second criteria to ensure success is that the compound must remain in solution for the duration of the study, because any precipitation can clog or block the exit port of the pump, resulting in incorrect delivery. The criteria for selecting a vehicle for use with ALZET pumps should include the solubility of the test compound, tissue compatibility, pump compatibility, pH considerations for compound stability, and of course, sterility. Here's a list of vehicles that are known to be compatible with ALZET pumps. In general, the pumps are compatible with aqueous solutions, dilute acids and bases, and low concentrations of DMSO and ethanol. The pumps are not compatible with natural oils and most organic solvents. This should be avoided since they will cause degradation of the pump reservoir and compromise the study. For compounds that are soluble in aqueous media, Ringer solution is an appropriate vehicle since it is sterile, particle-free, pyrogen-free, and non-irritating. And other popular options include saline, PBS, and artificial CSF for CNS applications. However, as you may already know or be aware, many experimental agents, especially small molecule therapeutics, are poorly soluble in aqueous solutions due to their hydrophobic nature. For successful delivery, these compounds must be dissolved in a non-aqueous vehicle that is not only compatible with the ALZET pump reservoir material, but also effective at preventing compound precipitation during the entire dosing period. This refined list of vehicles represents those vehicles that can be used to improve the solubility of hydrophobic compounds. In some cases, 2 or more of these solvents can be mixed together to prepare a vehicle combination with stronger dissolving properties. And here are some examples of vehicle formulations that have been used successfully with ALZET pumps for this purpose. Since DMSO is compatible with ALZET pumps at a maximum of 50% concentration, it can be mixed with polyethylene glycol, also at 50%, for stronger dissolving capacity. In other cases, ethanol can also be added to the DMSO PEG solution to improve the dissolving capacity even further. Another example is a study published in Drug Delivery. Researchers at Elan Pharmaceuticals and Pharmacyclics reported the development of a vehicle formulation containing a mixture of PEG 300, cremophor, glycofurol, ethanol, and propylene glycol. And this formulation was effective at solubilizing 2 of their investigational compounds that have poor aqueous solubility. The solvent formulation was fully compatible with the ALZET pump reservoir material, and the compounds remained soluble and chemically stable throughout the administration period. This vehicle combination also prevented compound crystallization when the drug solution was exposed to the aqueous environment at the site of its release from the pump, such as in vitro during priming of the pump or in vivo at the site of pump implantation. Another misconception is that high viscosity solutions can't be delivered with ALZET pumps. Well, the fact is that ALZET pumps are capable of delivering homogeneous solutions with a viscosity of up to 100,000 centipoise or millipascal second. And this is roughly 10 times more viscous than the thickness of auto engine oil, or approximately the viscosity of sour cream. So the forces generated by the pump, the osmotic forces, are great enough to deliver very, very viscous solutions. When it comes to making calculations about drug concentration, drug dose or pump duration, it is critical to use actual pump specifications rather than the nominal specifications listed on marketing materials, as I mentioned earlier. The nominal performance is only a target for all pumps manufactured. However, individual lots of pump will vary from this target within acceptable limits. The actual pumping rate and fill volume of a particular lot of pumps are listed on the top of the instruction sheet included with each box of pumps. The image here on the left shows an example of how this lot-specific information is listed on the instruction sheet. And this data should always be used for making calculations related to drug formulations. The table on the right shows the actual release rate and fill volume for ALZET model 2002 from this lot of pumps. And you can see how it varies from the nominal specifications. So when calculating the actual duration for this lot by dividing the fill volume by the release rate, you can see that the total duration for this lot is actually 16.1 days instead of 14 days. And more importantly, drug dose will also differ depending on the values that are used to make these calculations. The pump filling procedure is very simple and very straightforward. It is essential to use a sterile technique when filling the pump to avoid microbial contamination and infection after implantation. It is also important to use blunt end filling tube that is provided with the pump. If using a bevel needle, that can puncture and damage the reservoir. When filling the pump, a small amount of back pressure can be expected, and this back pressure can be more pronounced when filling with viscous solutions. So it is recommended to fill the pump slowly to avoid the formation of air pockets during filling. After filling, the flow moderator is inserted into the pump to the existing orifice. Before priming, the pump should be, before implantation, the pumps should be primed in saline at 37 degrees Celsius for a predetermined period of time. This priming step will enable the pump to work up to its mean pumping rate in vitro so that the pump is ready to release at a predetermined constant rate immediately after implantation. Recommended priming times for each pump model are available on our website, or you can contact us for that information. In some cases, in vivo priming might be acceptable also, as long as you're aware that the animals will not be receiving a full dose until the pump reaches a steady state. However, priming is highly recommended in the following cases. When immediate pumping is required, when using a catheter or brain cannula, when delivering a viscous solution, or when delivering a drug that may cause acute toxic effects. Once the pump is filled with drug solution and prime is ready for implantation, and the pumps, the ALZET pumps are designed for subcutaneous or intraperitoneal implantation, as shown here in this cartoon graphics. The usual site for subcutaneous implantation is in small rodents is on the back, slightly posterior to the scapula. The implantation procedure is relatively simple. Once the animal is anesthetized and shaved, the implantation procedure can be completed in less than a minute. You first make a mid-scapular incision and create a subcutaneous pocket by blunt dissection using a hemostat. The pocket needs to be just the right size to allow some free movement of the pump. If it is too large, it will migrate to the side of the flank. If it is too small, it can cause pressure necrosis at the site of implantation. After the pocket is made, you insert the fill pump into the pocket, and then the incision can be easily closed with wound clips or sutures. Here's a table describing the minimal animal size requirements for implantation of ALZET pumps. In general, the smallest pumps, the 100 microliter size pumps, can be implanted subcutaneously in animals as small as 10 grams, which is then it makes them ideal for use in mice. This size can also be implanted in the peritoneal cavity of mice, but the animals need to be at least 20 grams. The 200 microliter size pumps can be implanted subcutaneously in animals weighing at least 20 grams, and therefore they're also adequate for implantation in adult mice. They're not recommended for intraperitoneal implantation in this species. And the larger size pumps, the 2 milliliter sizes, can be implanted subcutaneously in rats weighing at least 150 grams, and they can go in a rat that is at least 300 grams for IP implantation. A common concern about ALZET pumps is to think that they can restrict animal movement. In fact, rodents have very loose skin on their backs, and they can accommodate the pump with no apparent discomfort. When implanted according to the animal size guidelines described earlier, the pumps should not impose significant impediment on the animal to perform normal activity. And in fact, ALZET pumps are commonly used in studies that involve behavioral testing, like the open field test, the Rotarod performance task, or the swim test. We collect this information in our database, and we can provide a list of published studies on request. Another benefit of ALZET pumps is that they can be connected to a catheter. This enables direct administration of compounds to almost any target area or tissue. This method also allows for intravenous administration or brain cannulation for delivery to the cerebral ventricles or brain parenchyma. The pumps can also be used with other specialized catheters for delivery to the spinal cord, to peripheral nerves, to tumor site, and other organs or tissues. A catheter can also be used to delay drug delivery, reduce drug waste, or deliver incompatible solvents. In the first case, the pump is filled with drug solution and the catheter is filled with vehicle. The pump works as normal by compressing the reservoir and delivering the vehicle solution from the catheter first. Once the contents of the catheter are displaced, the drug solution reaches the tip of the catheter, and then drug infusion begins. This method is commonly used to provide a surgical recovery period after pump implantation, particularly in brain cannulation applications. In the second and third case, the pump is filled with vehicle solution, and a length of catheter tubing is filled with the drug solution. This method can be used to administer a drug solution for a shorter period of time than the duration of the pump, in order to reduce drug waste. For example, if you plan to use a 7-day pump for delivery of a compound for exactly 4 days, without the catheter, the pump will need to be filled completely with drug solution, and 3 days worth of solution would be wasted. The catheter can also be used to deliver solvents that are incompatible with the drug reservoir, but compatible with the catheter. A common question from researchers is how to verify that the delivery using ALZET pumps. One way to confirm that is the pump is working is to evaluate blood levels of the drug at several time points during the course of the study. In the event that determination of circulating blood levels is not possible or technically undesirable, users should measure residual volume in the reservoir when the pump is removed from the animal at the end of the study. This provides a quick and easy confirmation of delivery. You should use the same blunt end filling tube initially used to fill the pumps to aspirate the remaining solution from the pump reservoir. The residual volume can be compared to the theoretical volume that would remain in the pump based on the actual pump release and time that is implanted in the animal. A common misconception, though, is to weigh the pumps at the end of the study to confirm delivery. You should know that the weight of a partially empty or discharged pump cannot be used to determine the quantity of drug delivered. And the reason is that the pump absorbs water during operation. Therefore, the weight of the pump at the end of the study will be equal, or it might even be higher than its weight at the start of the study. And cutting the pumps open is also not a reliable means for verifying delivery, since the contents of the hydrated salt solution, the hydrated salt layer, are often mistaken for drug solution. So The best way would be to measure residual volume, and that provides a very quick, easy, and qualitative means of confirmation. So now I will shift my focus to the iPRECIO programmable pump. And the iPRECIO pump is an advanced infusion device for drug delivery applications in mice, rats, and larger animals. This pump provides greater flexibility than the ALZET Osmotic Pumps in terms of changing flow rates and durations and other capabilities. Complex dosing protocols can be programmed with the dedicated software with a computer, and then the protocol is then downloaded to the pump. The two iPRECIO pump sizes or models available are shown here with their corresponding dimensions, and I'll go over those in greater detail. So the three main features of the iPRECIO pump is that it is implantable, refillable, and programmable. The iPRECIO pump is designed so it can be fully implanted subcutaneously in mice, rats, or larger laboratory animals. The SMP 200 pump can be implanted in rats or larger animals weighing at least 230 grams. The smaller iPRECIO SMP 300 pump can be implanted in mice that are at least 22 grams. Once implanted, animals are unrestrained and can move freely in their cages during drug infusion, just like the ALZET pumps. And this feature allows for group housing of animals, which in addition of reducing animal handling, they help reduce stress and improve overall animal welfare. Refilling of the pump is not possible with the ALZET pumps, as you know. However, a unique benefit of the iPRECIO pump, its ability to be refilled after implantation while inside of the animal. Test solutions can be exchanged or replenished via the percutaneous access to the pump through a resealable septum. This feature enables researchers to perform chronic infusion studies without having to replace empty pumps, which eliminates the need for multiple surgeries during the course of the study. The ability to refill the reservoir is also helpful when testing multiple drugs or when working with drugs that have very poor stability or poor solubility that require to be formulated or prepared fresh for dosing very often. Although the iPRECIO pump is refillable, a common misconception is to incorrectly assume that it can be reused. And just like the ALZET pump, the iPRECIO pump is designed to be a disposable product for single use. It's important to note that the battery life is the main limiting factor preventing the iPRECIO pump to be reused. Sterility and the potential for cross-contamination is also another issue to consider. Another unique feature of the iPRECIO pump is its ability to be programmed to deliver according to the needs of the study. This allows full control and flexibility for dosing animals. You can set your own infusion protocol by entering the start and stop time, the desired infusion rates, the duration of administration, and all the necessary study details. The complete infusion protocol is then downloaded to the pump's flash memory via infrared communication for the SMP SMP 200 pumps, or by wireless communication for the SMP 300 pumps. Once implanted, the pumps will deliver according to the programmed infusion profile. This slide shows some examples of complex dosing profiles that can be fully automated using iPRECIO pumps. This includes constant and variable dosing, delayed delivery to provide a surgical recovery period after implantation, or studies requiring the administration of multiple drugs that need washout periods in between drugs, dose escalation studies to study dose response in a single animal, or dose adjustments to account for the growth of the animal during the course of the study. It also can be used for chronic bolus dosing or circadian rhythm infusion. So all these infusion or dosing protocols can be automated using the iPRECIO pump. The key components of the iPRECIO pump are the accurate and reliable rotary finger mechanism, which I will describe later, the microprocessor and the associated circuitry for the pump control, power source or battery, the fluidics components, which include the reservoir, catheter, and the filling port, and the associated software and base station for programming. This slide shows images of the S&P 200 pump describing all of its components. The top view shows the pump reservoir, which can hold 900 microliters of solution, the filling septum, the two suture anchors that help keep the pump in place after implantation, the inner and outer catheter, and the rotary finger mechanism composed with the micromotor, the cam, and 7 finger points. The bottom view on the right shows the locations for the battery, the CPU circuit, and the infrared communication. And the parts for the S&P 300 pumps are very similar to those of the S&P 200 pump, with some exceptions, obviously. The S&P 300 pumps have an antenna for wireless communication, and they also have a power switch, as shown in the bottom view image on the side. This is an important improvement over the S&P 200 pump that helps conserve battery life before use. The top view image shows the septum port and the septum port for refilling and filling, and the pump reservoir also, which holds 130 microliters. So let's go over how the iPRECIO pump works to administer solutions as programmed. The technology driving the infusion is a patented rotary finger method. This method is a unique form of peristalsis. Once the pump is activated, a micromotor slowly revolves in a clockwise direction, turning the cam with its forward projection. In each quarter rotation, a single cam projection sequentially pushes up each of the 7 finger pins. This continuous cycle compresses the liquid-filled catheter, creating a peristaltic-like movement of the fluid. As the solution moves through the tube, it is expelled from the pump reservoir into the animal. This unique method provides accuracy better than plus or minus 5%. And for additional confidence, each pump is tested and calibrated at factory to ensure that every pump works properly. For programming the pumps, you will need to purchase the iPRECIO management system, which includes the data communications device, USB or LAN cable, the software installation CD, and the user manual. And this is separate from the pumps, from the actual pumps. Programming the pumps is relatively straightforward once you familiarize yourself with the program. And the program guides you through a series of windows or screens where you enter relevant study information. In the first screen, for example, this is dedicated to the general study details. Here's where you enter information about the study ID, the date, the animal species, compound, vehicle, routes of administration, and other information relevant to the study. The second window is where you enter the concentration of the solution, the minimum and estimated maximum animal weight that is expected at the end of the study. The software will then automatically calculate the minimum and maximum dose, which can be administered during the experiment. You can also select an infusion mode and input the dose, or to input this dose, or select a flow rate mode and enter the desired flow rates that you want to infuse. In the third screen, you program the infusion protocol for each animal and enter individual animal ID, body weight, sex, and other animal information. In this step, you also set the start and end date and detect the pump. The last screen is dedicated to individual pump programming. It's worth mentioning that that up to 10 flow rate steps can be programmed in variable infusion mode. And even further sophistication can be obtained by also using a repeat mode, where 10 steps can be programmed within up to 5 repeat cycle loops. And each loop may be repeated 250 times. Therefore, the iPRECIO pump is capable for programming with extremely complex dosing parameters, and in many cases the capability would not even be fully utilized for most dosing studies. The program also contains a monitor function or a status manager window that allows you to confirm the administration schedule and the remaining volume of the pump reservoir during the course of the study. It also provides information about the refill dates and other information that you can follow during the course of the study. Here's a table comparing the main features of both iPRECIO pumps. I've already described most of these features earlier, but I should add that the iPRECIO SMP 300 pumps can be programmed wirelessly with flow rates of up to from 0 to 10 microliters per hour, and that's with a 0.1 microliter per hour resolution. And the maximum duration is up to, that can be achieved, is up to 47 days. The SMP 200 pumps are programmed by infrared communication with flow rates up to 30 microliters per hour, and the maximum duration possible is 6 months at the low flow rates. In fact, the duration of the pump is dependent on battery life. The SMP 200 pumps can deliver up to 6 months when programmed to deliver at 1 microliter per hour. As you can see in the table, the battery life is significantly reduced with the faster flow rate. So at 30 microliters per hour, you get about 1 week of battery life. The battery life on the SMP 300 pumps is not only affected by flow rate, but also by the communication interval that is selected. These pumps give you the option to program a communication interval, which is communication with the base station, and you can program that to communicate every 2 hours, every minute, every 2 hours, every 6 hours, 24, or none. And in order to maximize battery life, what we recommend is to program the pump to communicate with the base station every 6 or every 24 hours for a given flow rate. At that point, the battery life is conserved the maximum, and you still get constant communication with the base station. Vehicle compatibility is very similar to that of the ALZET pump, with some exceptions. The list here in the left represent those vehicles that are not compatible with the iPRECIO pump. Other vehicles, particularly viscous solutions, can only be used for short-term studies or studies where refilling is not needed. The finger point technology of the pumps is not influenced by the viscosity of the solution. However, the viscosity, higher viscosity, it makes it very difficult to fill and refill through the septum with a 27 gauge needle, and that's where the complication is. Viscosity up to 20 centipoise has been evaluated, and it's acceptable. Here's a short list of some of the agents that have been used with iPRECIO pumps. Obviously, this is a short list. There are many other ones that have already been used. Research with these pumps continues to grow, and we can provide a list of publications for your information. And this slide shows various images that describe the filling and refilling procedure for the SMP 200 pump in a rat. The pump is filled with test solution via the percutaneous access to the pump. through the refill septum as described earlier. This refill septum can retain its integrity even after more than 50 needle insertions, which is more than enough for any study. The reservoir is elastic and with a geometry that allows for gentle palpation to approximate the level of fluid that is in the reservoir, so that can be felt through the skin. These pumps can be used for subcutaneous and intraperitoneal implantation and delivery of agents, as shown here in these graphs. They can also be used with specialized catheters for intravenous administration. Here showing some cartoons for the SMP300 in mice and the SMP200 in rats. In some cases, if you're using specialized catheters with modified tips for delivery into vessels, you can join the catheter from the iPRECIO pump to that special catheter via a coupler or a catheter connector. They can also be used for CNS applications where they can be connected to an intrathecal catheter for delivery to the spinal cord, or to a brain cannula as shown here for administration of agents to the cerebral ventricles or brain tissue in rats or mice. The iPRECIO catheter can be connected directly to the brain cannula that is included in the ALZET brain infusion kits. Or if needed, it can also be used with a coupler if other materials are used. So in conclusion, implantable pumps are a convenient and reliable method of drug delivery, and they offer significant advantages over conventional dosing methods and external pumps. For most research applications, ALZET pumps offer a simple method for continuous and reliable dosing of animals. However, when complex dosing is required to perform a study, the iPRECIO programmable pumps may be best suited to meet those demands. This table shows a brief comparison of both products to help you determine which pump is best for your specific study. Obviously, this does not include all the requirements that need to come into play when choosing a study. We can help with that decision. That decision is not always simple, but perhaps we can provide resources for you to help you. We have a number of resources including surgical implantation videos, protocols, publications that can be helpful for your study. So here's our contact information. Our website is also a great resource, and I would like to thank you for listening and your attention. It's been a pleasure to provide you share this information with you, and now I will transfer this to Martin for a Q&A session. And if you have any questions, feel free to post those in the chat box. Thank you very much.
Start by asking, this has asked a few times, specifically Klaus has asked, is it possible to use two pumps in parallel? And others have asked, how many pumps can be implanted in a single animal? How would you answer that question?
Okay, yeah, thank you, Klaus, for that question, and everyone. Yeah, that's a very good question. We get that asked a lot. Yes, the answer with the ALZET pumps particularly, you may be able to use more than one pump, but that ultimately is dependent on the size of the animal. So, for example, we do have certain parameters and guidelines, as I showed earlier, and we like to stick to those guidelines. So, for example, if you're working with a 20-gram mouse, the 200 microliter size pump is adequate. but you could use 2 of the smaller pumps. In larger animals, you may get away with using, the 200 microliters. So if size in rat, for example, you can use multiple pumps. We do have references on our database that show multiple pumps being used in different species. We'll be glad to provide that information to you. So the main requirement is really to stick to the number of pumps that would still retain, animal welfare, and the animals will not be affected by that. But certainly, multiple pumps can be used.
Okay, great. Thanks for that answer. I'm sure that addresses the question. Another question is, what is the smallest drug suspension volume that can be incorporated into the pump? It's not specifically mentioned here whether it's ALZET or the iPRECIO, but how would you answer that question?
Yeah, I'm glad that question was asked also, because that is another misconception that I didn't cover on my presentation. But many times, this, and I believe this is particularly related to the ALZET pumps. As I mentioned earlier, we have 100 microliter, 200 microliter, and 2 milliliter pumps, and sometimes researchers just, have half of the solution that they want to administer, and incorrectly fill the pump halfway, for example. The straightforward answer is that the pumps need to be filled completely up to their maximum volume for effective delivery and for continuous delivery. As described in the presentation, the ALZET pumps have a fixed volume reservoir, so it's not an elastic. It doesn't fill up and expand as you're filling. So it's just a fixed volume. So if you, for example, are using a 200 microliter pump and you fill it with 100 microliter, well, you'll end up with basically 100 microliters of air. So when the pump is implanted, at some point, when that air makes its way into the flow moderator and out through the pump, then that will affect continuous delivery of your drug. So because you'll be delivering air. So the answer is that they need to be filled completely. So there's no the minimum volume that they can be filled with would be its actual volume. With the iPRECIO pumps, it's not an issue, because the pump can be the reservoir is flexible, and so it can be filled with less volume than the actual reservoirs. For example, on the S&P 200 pump, which has a 900 microliter reservoir volume, you don't necessarily need to fill it completely with 900 microliters. You can fill it with half of that, for example.
Okay, great. You did present a couple slides on how to measure the residual volume at the termination of a study. Can you just, there's been a few questions about that. Would you just elaborate then, what is the best way to determine how much solution is left in the pump after an experiment?
Sure, yeah. Yes, I did cover that, Martin, but I think that it was, I covered it in very briefly. So the residual volume, at the end of the study, that would provide a very quick assessment of pump functionality. Basically, you just take, when you remove the pump, remove the flow moderator, aspirate, use the same filling tube that you used to fill the pumps at the beginning of the study, and aspirate whatever solution remains in the pump at that period of time. You need to be careful to try to remove as much as the actual volume as possible, so that you can at least have a way to measure that. It won't provide a quantitative assessment. It would be more like a qualitative assessment of functionality. You know, you'll get an approximation of how much was delivered and how much wasn't. So you compare that volume, that residual volume, to the volume that should have been delivered based on the actual specifications of the pump. One thing to note also is that when making the residual volume calculation, you should know that 5% of the reservoir volume cannot be delivered. So even once the pump is fully compressed, it will still retain about 5% of the total volume. But that's not an issue in terms of pump performance. It's just because the pumps usually have a larger reservoir than what's needed for the total duration.
Okay, great. Let's ask one more question. Renee has asked a few, but one of the questions is, you talked about stability, chemical stability. Do the manufacturers of the drug compounds know the chemical stability of each compound, or is it dependent on the solvent that's used?
Yeah, it's a good question. A lot of times you can get that information, yes, from the manufacturer. The issue is that sometimes they provide stability under specific conditions that may not be the same conditions that you are using it for. So yes, in some cases the solvent that you use will impact the stability of the compound, and in fact many times different solvents are chosen for that specific purpose, which it is to enhance the stability of the compound. So okay. So yes, it is. And we also keep that information in our database for some drugs. So we, you know, almost every publication that comes out, we read it and we index it and add it to our database. And stability is one of the keywords that we look for in terms of keeping that information, so that when customers have specific questions about that, we can do a search for that and provide that information.